Hi, my name's Sarah and I'm going to be demonstrating the Quadrant streak. So before I start, I'm actually going to talk about lab safety. So normally in a lab, I would be wearing a lab coat and safety goggles, as well as closed-toed shoes, and my hair would be tied back and out of the way, as well as no excessive long jewelry. And then before I start, I'm also going to be washing my hands in the sink with proper technique. Make sure to scrub with soap and water for 15 seconds, dry it off completely. Now that we've gone over lab safety, I can go ahead and start demonstrating. The first thing I'm gonna do is label the agar plate, and this I'm gonna actually be doing, this is my agar plate, and I'm actually gonna be labeling it on the bottom of it, not on the lid, but on the bottom side of the agar plate on the edge. And I'm gonna go ahead and write my name, today's date, and then I'm also going to include the type of bacteria that we're going to be doing. So now that the plate is labeled, I can flip it over and open it. And this right here is actually going to be the plate with the colony bacteria that I'm drawing from. So now that I have the inoculating loop in my hand, I've grabbed it from the rack. This is my Bunsen burner. I'm gonna burn it over a flame to sterilize it. Then I'm going to dip it into the colony agar plate, and I'm gonna draw it on my own. I'm gonna start with the first quadrant and streak it like this, so it has a zigzag pattern. Now that I've streaked it, I'm going to go ahead and sterilize it again. And now that it's sterilizing, I'm going to go ahead and start with my second quadrant. And I'm just going to draw from the first and create a zigzag like this. And then I'm going to sterilize it again. And then I'm going to go ahead and start with the third quadrant and pull like this. So it'll look like this. And then I'm going to sterilize it once again and then start on my last quadrant. That's going to look like this. So now that I have all four quadrants pulled, I'm gonna burn the inoculating loop to sterilize it and put it away. And then I'm going to close this and the colony, make sure they're nice and closed, and I'm gonna put this agar plate in an incubator for 24 hours so the next day I can go and see what I've grown. Thank you.